Acetyl-L-carnitine seems to be a powerful immunomodulator in the course of Mycobacterium tuberculosis and other mycobacteriosis. Resveratrol belongs to a class of natural compounds called stilbenes. The chemically synthesized analog, azastilbene, was tested against in vitro Mycobacterium tuberculosis infection and was found to have anti-TB activity comparable to pyrazinamide. Glycerol monolaurate from virgin coconut oil was comparable to the effectiveness of the anti-tuberculosis drugs streptomycin, isoniazid, rifampicin, and ethambutol. Glycerol monolaurate proved effective to one bacterial strain which is resistant to isoniazid. The University of Maryland Medical Center recommends TB patients take alpha-lipoic acid, CoQ10, and resveratrol daily for antioxidant support. Immunologists at Harvard discovered that people who drank 5 cups of black tea a day for 2 weeks transformed their immune system into Hulk cells that pumped out 10 times more cold and flu virus fighting interferon proteins that defend against infection than did the immune system of those who didn't drink black tea. The immune boost is caused by L-theanine, an amino acid common to black, green, oolong, and pekoe teas. 20 ounces of tea a day produced a 10-fold increase in gamma-delta T cells, and a similar consumption of coffee afforded no such protection. Not just the common cold and flu, even diseases such as tuberculosis and malaria. We think the interferon boost from tea may help prevent or lessen the severity of all these conditions. A 43-year-old man drank 16 cups of Earl Grey tea a day and had muscle cramps and blurred vision. His symptoms went away when he drank 4 cups a day. The essence of bergamot, the aromatic flavor of Earl Grey tea, caused the muscle cramps and blurred vision. The essential oil of bergamot enhances the levels of amino acid neurotransmitters in the hippocampus of rat. Parkinson's disease is associated with hippocampal atrophy. 5-methoxyseralin is an extract of bergamot oil. 5-methoxyseralin increases the plasma melatonin levels in humans. After 7 days of crude green tea extract therapy, oxidative levels of tuberculosis-infected mice went back to near normal, with a concomitant increase in glutathione levels. There is high oxidative stress during early stages of tuberculosis, and antioxidants such as green tea extract can play a vital role by reducing stress through adjuvant therapy. University of Central Florida researchers discovered nicotine stopped the growth of Mycobacterium tuberculosis in laboratory tests, even when used in small quantities. Nicotine can be oxidized to nicotinic acid, niacin, which is present in minute amounts in all living cells. The corresponding amide, nicotinamide, is an essential B vitamin. Ibuprofen slowed the lesions of tuberculosis in the lungs of mice, and lived longer than mice not treated with ibuprofen. In a Turkish study, melatonin showed a broad antibacterial spectrum against all the studied bacteria, which included Streptococcus, Staphylococcus, E. coli, Pseudomonas aeruginosa, and Salmonella. It is concluded that melatonin showed antibacterial effects against multi-drug-resistant tuberculosis. Scientists in South Africa showed that melatonin, when used with the frontline TB antibiotic isoniazid, potentiates isoniazid and causes a 3-fold increase in the efficacy of the drug in killing multi-drug-resistant tuberculosis. Melatonin is also an effective iron chelator and scavenger of hydrogen peroxide. Isoniazid, rifampicin, and pyrazinamide are all toxic to the liver. A flavonoid complex extracted from the seeds of the milk thistle plant has been used to treat liver ailments for over 2,000 years. In Study 1, curcumin, silymarin, and N-acetylcysteine were shown to be hepatoprotective and need to be further explored as a powerful adjuvant therapy along with anti-tuberculosis drugs. In Study 2, silymarin protects liver against toxic effects of anti-tuberculosis drugs in experimental animals. New TB patients will be given standard drug therapy supplemented with placebo or silymarin at 140 milligrams 3 times a day for 4 weeks to see if it can prevent liver damage. Silymarin reduced the incidence of anti-tuberculosis drug side effects on the liver. Silymarin and melatonin offer neuroprotection against MANAB and paraquat-induced Parkinson's disease by the modulation of oxidative stress and planned cell death. Ocimum sanctum, also called holy basil or tulsi, is a sacred plant in Indian medicine. Ocimum sanctum has a potent anti-Mycobacterium tuberculosis effect, both directly and in infected macrophage culture. Co-administration of Ocimum sanctum leaf extract, along with anti-tubercular drugs, significantly prevented liver damage in rats. Multiple studies have shown the neuroprotective effect of Ocimum sanctum in various models of Parkinson's disease. Hops have a long history of use as a natural preservative in beer. A little-known benefit, however, is that hops also contain high concentrations of unique alpha and beta acids that can inhibit the growth of gram-positive bacteria at surprisingly low concentrations. Natural killer cells are innate lymphocytes, which are a first line of defense against infection and cancer. Naltrexone treatment increases NK cell cytolytic activity and cytokine production in the spleen within the living. Natural killer cells play a crucial role in innate defense mechanisms against M. tuberculosis infection. Furthermore, NK cell functions are dependent on adequate levels of glutathione. Glutathione levels are significantly reduced in peripheral blood mononuclear cells and red blood cells isolated from tuberculosis patients. Glutathione, an important antioxidant, requires magnesium for its synthesis. A study compared the magnesium levels of 40 pulmonary tuberculosis patients versus 25 healthy controls. The study showed that serum magnesium levels were significantly low in patients with pulmonary tuberculosis in comparison to controls. Professor Delbay also performed experiments with the internal applications of magnesium chloride and found it to be a powerful immune stimulant. In his experiments, phagocytosis increased by up to 333%. Dendritic cells are the sentinel cells of the immune system. They initiate immune response and are known to orchestrate interactions of more than a dozen types of immune system cells. Two of the cells dendritic cells orchestrate are T cells, a type of white blood cell, and interferon gamma. Interferon gamma is a small protein called a cytokine and is the principal mediator of macrophage activation against pathogens. DHEA-enhanced Mycobacterium-specific T-cell proliferation and interferon gamma production after being induced by M. tuberculosis-stimulated dendritic cells. Mice infected with tuberculosis were then treated with DHEA and its derivative, beta-androstenediol. Both compounds were protective, particularly beta-androstenediol, which caused a fall in bacterial counts and prolonged survival. Androstenediol for tuberculosis and androstenediol for Parkinson's are metabolites of DHEA. A synthetic form of androstenediol, HE3286, protected mice from the neurotoxin MPTP and reduced inflammation, significantly improved motor function, increased tyrosine hydroxylase positive cells, and decreased the number of damaged neurons. While certain confirmatory studies remain in progress, our data to date suggest a potential role for HE3286 in the treatment of Parkinson's disease and Parkinsonism-related disorders, and provide incentive for further investigation.